18. A contaminated vaccine leads to cancer-breaking genes. On April 12, 1955, following nationwide clinical trials on 1.8 million American schoolchildren, Dr. Thomas Francis Jr. of the University of Michigan announced that the Salk vaccine against polio, at the time the leading crippler of children, was safe, effective, and potent. Euphoria swept the country. As Richard Carter noted in his biography of Jonas Salk, people observed moments of silence, rang bells, honked horns, blew factory whistles, fired salutes, kept their traffic lights red in brief periods of tribute, took the rest of the day off, closed their schools, or convoked fervid assemblies. Therein drank toasts, hugged children, attended church, smiled at strangers, forgave enemies. Up until 1961, the culture for preparing the life-saving polio vaccine used kidney cells from rhesus monkeys from India. When a measles infection from human contact spread throughout the monkey colony, Merck switched to using African green monkeys. Such an event might seem unfortunate, but what happened next is another example of how serendipity works its magic. Thorough testing uncovered the surprising finding that the first monkey species harbored a previously undetectable virus that was harmless to them but capable of killing and replicating in the cells of the second species. The conclusion was both startling and distressing. The Salk polio vaccine, in use since 1953, was badly contaminated with a virus. Had it not been for the chance measles infection and the subsequent switch to African green monkeys, the virus would not have been detected. Alarm spread when both Merck and NIH researchers showed that the virus caused cancer when injected into newborn hamsters. The shocking reality could not be dismissed. The polio vaccine, which acted to reduce or eliminate poliovirus epidemics all over the world, was contaminated with a virus that initiated cancer in hamsters. Mass immunizations with injections of 3 doses of vaccine were so popular that about 450 million doses had been administered in the years 1955 to 59. The United States Bureau of Biologics acted swiftly to eliminate the virus from poliovirus seed stocks and grow poliovirus solely in the African green monkey cells. Meanwhile, amid the intellectual ferment in molecular biology of this era in cancer research, Arnold Levine, a molecular biologist at Princeton University, was intrigued by this newly discovered virus shown to transform normal cells into cancer cells. Levine started by thinking that the virus's protein shell might induce an antibody in the blood. The antibody could be detected as a biomarker to indicate the presence and severity of the infection and even the results of treatment. Levine's search led him in 1979 to unexpectedly discover a new protein in the blood. He named it simply P53 because the protein has the molecular weight of 53,000 hydrogen atoms. For 10 years after its discovery, Levine followed the P53 gene and its protein down a false trail, believing it to be an oncogene, a cancer-accelerating gene. He found that putting the gene into normal cells, using rat embryos, made them cancerous. In 1983, he cloned the gene for the first time and demonstrated what appeared to be its tendency to induce tumors. But Levine was wrong about the actual nature of P53. Finally, in 1989, the real breakthrough occurred. While pursuing the genetic causes of colorectal cancer in humans, Bert Vogelstein at Johns Hopkins University found a mutation in the P53 gene. As Levine reviewed the data, he experienced what can only be described as a eureka moment. In a conceptual leap, he realized that P53 was not a gene that stimulates cancer, but a gene that suppresses tumors. He had been led astray because the clones he had used in the early experiments were unwittingly composed of mutants. It was as though he was struck by lightning when he came to understand that the P53 gene had to be mutated in order to foster cancer transformation within a cell. That is, its normal function is to inhibit division and growth of tumor cells. The normal P53 protein acts as an emergency brake to arrest any runaway growth of cells that may have acquired cancerous tendencies, and as a damage control specialist to induce cell death under specific circumstances, such as in the presence of DNA damage. It has been dubbed the guardian of the genome because of its vital biological functions. After 10 years of dedicated research, suddenly, Levine ruefully noted, P53 became a hot ticket in cancer research. A single mutation in the 135th position of its 393 amino acids can eliminate the surveillance capability of the protein and allow a cancer to grow. In its mutated form, it is found in more than 50% of human cancers, including most major ones. Among the common tumors, about 70% of colorectal cancers, 50% of lung cancers, and 40% of breast cancers carry p53 mutations. The p53 gene is also linked to cancers of the blood and lymph nodes. Put another way, of the 10 million people diagnosed with cancer each year worldwide, about half have p53 mutations in their tumors. The gene's inactivation through mutation enables tumor cells, in a famous phrase, to reach for immortalization. In 1993, Science magazine named p53 the molecule of the year. It has generated much enthusiasm for the development of strategies for the diagnosis, prevention, and cure of cancers. Arnold Levine is a round-faced, high-energy, fast-thinking, fast-talking individual who commands deep affection from his laboratory team. He went on to become the president of Rockefeller University in New York. Would he consider the discovery of p53 serendipitous? Absolutely, he frankly responds, noting the frequency of chance discoveries and citing the observations of Richard Feynman, the Nobel laureate in physics, on how scientists commonly write their articles in such a way as to cover their tracks when it comes to how they stumbled upon the truth. Probing the genome for the renegade pathway. A major focus resulting from such advances in recognizing the influences of genetic mutations upon cancer is the field of targeted drug therapy. In the human cell's intricate inner circuitry are dozens of molecular chains of communication, or signaling pathways, among various proteins. It is now understood that there are roughly 10 pathways that cells use to become cancerous, and that these involve a variety of crucial genetic alterations. Research is actively directed toward developing drugs able to interfere with the molecular mechanisms that drive the growth in a tumor and that are attributable to these mutated genes. A handful have been approved for use. The best known is Gleevec, which has a dramatic effect on an uncommon kind of leukemia called chronic myelogenous leukemia, CML, and an even more rare stomach cancer, gastrointestinal stromal tumor, GIST. CML, however, is exceptional in that it is due to a single gene mutation, and the drug is able to block its specific tumor signaling mechanism. Unfortunately, over time, further mutations circumvent the molecular signal that Gleevec blocks, building drug resistance. Most cancers are more complex, with multiple mutations that require a multi-pronged attack. Picking the right targets is critical, and the most promising new diagnostic technology is the DNA microarray, or gene chip. Gene chips can identify mutations in particular genes, as well as monitor the activity of many genes at the same time. It is hoped that this precise molecular biology will lead to the design of a range of drugs that target highly specific signaling pathways of cancer. Cancer was long thought of as one disease expressed in different parts of the body: breast, lung, brain. But researchers are now teasing apart the myriad genes and proteins that differentiate cancer cells, not just from healthy cells, but also from each other. The aim is to classify cancers mainly by their genetic characteristics, by which pathway is deranged, not by where in the body they arise or how they look under a microscope. The ideal is to fine-tune the diagnosis and treatment of cancer for more personalized medicine. But knowledge of how to do that is still lacking in most cases. The magic bullet has yet to be found. Many of the new cancer drugs are targeting things in cancer cells that may or may not be driving that cancer. The drugs in use by many patients in clinical trials work for only a minority. In truth, scientists cannot really identify, in most cases, the Achilles heel of a cancer cell or understand very well the targets at which they are shooting. And as the tumor grows, the target is ever changing. The successes claimed seem at times to be merely instances of drawing a bullseye around the spot where the arrow landed. Even here, serendipity may be grafted upon imperfect knowledge. The story of the drug Iressa illustrates the point. Iressa blocks the activity of an enzyme that stimulates cell division. As the enzyme is overabundant in 80% of lung cancers, researchers hypothesized that the drug would be an effective treatment for most lung cancer patients. They were both disappointed and perplexed when only 10% had a strong response. It then became evident that the patients who did respond well not only had lots of the enzyme, they also had a mutant form of it. By contrast, those patients who had either no response or a partial response had only the normal version of the enzyme. In the recent case of a promising experimental drug targeted against melanoma, Serendipity played a major role. The drug, developed by the German company Bayer and by Onyx Pharmaceuticals, a California biotechnology company, goes by the awkward code name BY439006. The drug is the first to block a protein called RAF, one of a family of enzymes called kinases, which relay a cascade of signals leading to cell growth. The drug is most effective in kidney cancer, but not mainly by blocking RAF. Thanks to dumb luck, in the words of Dr. Frank McCormick, founder of Onyx and director of the Cancer Center at the University of California at San Francisco, it turns out that the drug also blocks a protein involved in the flow of blood to the tumor. In another discovery unforeseen by Bayer and Onyx, scientists in Britain found that RAF was mutated in about 70% of melanomas. When combined with chemotherapy, BY439006 shrunk melanomas in 7 of the first 14 patients. In his 1998 book, One Renegade Cell, Robert Weinberg, a pioneering scientist at MIT, put these discoveries and potentials into perspective. Until recently, the strategies used to find the genes and proteins that control the life of the cell have depended on ad hoc solutions to formidable experimental problems, cobbled together by biologists who lacked better alternatives. Time and again, serendipitous discoveries have allowed new pieces to be placed in the large puzzle. Discoveries of critically important genes often have depended on little more than dumb luck. Those who engineer these successes, technologies, gene mapping of cancers, targeted drug therapies, will view the discoveries of the last quarter of the 20th century as little more than historical curiosities. We have moved from substantial ignorance to deep insight. Nevertheless, can one doubt that serendipity will continue to play a role?